So good afternoon, everybody. It's a pleasure for me to share with you the result of the first chapter of my PhD thesis, which summarizes 1 year of field sampling in Côte d'Ivoire in order to explore the biotic and abiotic factors that influence the cucurbit infesting fruit flies damages and diversity. So to start with, in ecology, we assume that the distribution of a species is regulated by different factors, ecological factors, such as the climate, but also the dispersal capacity of the species, the biotic interactions with other species, and the nature and the structure of the habitat. And in addition, human activities such as agriculture is also disturbing all of these factors and influencing the final distribution of species. So the species of interest for me were species from the family Tephritid that you already know, and especially the species infesting cucurbits, because it's a very diverse family of pests worldwide that is threatening many crop, fruit and vegetables in the world. And my field study was Côte d'Ivoire, which is a West Africa country with a tropical climate that is favorable for many pests, and also with a lack of technical support in market vegetable production. The farmers massively use and misuse pesticides, which are not very adapted to the life cycle of fruit flies. That's why it's important to have other alternative control methods for this pest. So there are many alternative control methods that are more or less specific to a species, to a phenological stage of the flies, and to a sex also of the fly. But prior to implement any control method, it's important to identify the species of the pest, and to know some things about its behavior and life cycle. So that's what my study aimed in Côte d'Ivoire, with 2 research questions. So the first, it was to evaluate the importance of the cucurbit infesting Tephritid damages and diversity, and then to know and to understand what biotic and abiotic factors were influencing these damages and diversity. So to do so, I visited cucurbit plots from north to south Côte d'Ivoire in 4 in 4 zones from north to south. I interview also the farmers to know their agricultural practices, and I collected a few fruit that I bring back to the lab for incubation to then see and count and identify the fruit flies that would emerge, and to take some fruit characteristics. So I gathered 58 potential explanatory variables to try to explain my responses variables. So for the damages at the plot scale, I counted the number of fruit damaged. And at the fruit scale, I counted the number of pupae that recovered from every fruit. And to characterize the diversity, I counted the number of different species in each plot and in each fruit. And also I could calculate then the specific and the relative abundances of each species at the plot scale and at the fruit scale. Then for the data analysis, I started with multivariate analysis to see, to visualize where my potential explanatory variables were located in distance to the response variables, and I selected the ones that were the nearest to put then in a generalized linear model. And then I performed a step-by-step variable selection to finally retain only the variables that had an effect, a significant effect on my responses variables. So now for the results, for the damage, 90% of the plots I visited were infested, with infestation rates that ranged from 1 to 100%, and a mean of 31% infested fruit at the field scale. And for the fruits, 55% of the fruit I collected were actually infested by fruit flies, and they were hosting from 1 to more than 200 pupae per fruit, with a mean of 16. And for the diversity, I was able to identify 5 species of cucurbit-fruit flies, with the 3 main species being first Dacus ciliatus, which was present in 76% of the plots, then Zygodacus cucurbitae, which is an invasive species from Asia, and then Dacus bivitatus, which is like Dacus ciliatus, indigenous from Africa. So I will not go into the details of the stats, but in the end, over the initial 58 variables I had, only 5 had a significant effect on field damages, so on the proportion of infested fruits. So I found more damages in the plots that had a high weed cover, that were located in the north rather than in the south, that also display a variety of cucurbit that had a soft skin, and more damages in crop that at the end of the crop cycle. But I found less or lower damages in the plots that have been recently treated with one family of pesticide. And for the damages on the fruit scale, I found that there were more pupae in the fruits that had more lesions, that were more attacked. Also more pupae in zucchini rather than in cucumbers, which were the two cucurbit species that I compared. I found also more pupae in young fruit rather than in mature fruits, and in curved fruits rather than regular fruits. And yes. Then on the species diversity, at the plot scale, I found only 2 variables that had a significant effect on this on the species diversity, but which had the same signification, which means that the diversity was more important in plots that were less frequently treated. And at the fruit scale, I found the same variables that had the effects on the number of pupae, which means that the more pupae there is in a fruit, the more likely it is that the fruit was infested by many species, different species, in the same fruit. Now on the specific abundances at the plot scale, I found more individuals of Dacus bivitatus in the plots that were less frequently treated, and I found more Zygodacus cucurbitae in the plots that were the most infested, which has also the highest weed cover, as we've seen at the beginning, and which were located in the north. And at the fruit scale, I found more Dacus bivitatus individuals in the fruits that were more attacked, and I found more Dacus ciliatus individuals in zucchini and in curved fruit, rather than in cucumber and in regular fruits. And I found more of all individuals of all the 3 main species in the young fruit compared to the mature fruits. Finally, on the relative abundances, at the plot scale, I found that Dacus bivitatus was dominant in the plots that haven't been treated for more than 15 days. Also, Dacus bivitatus was dominant in larger plots and during the rainy season. And I found that Zygodacus cucurbitae was dominant in the more damaged plots. And finally, at the fruit scale, I found that Dacus ciliatus was dominant in the zucchini and in the curved fruits, and that it was in competition with Zygodacus cucurbitae in young fruits. So, to make this long story short and to try to make links between all of these variables, we concluded that plots with high weed cover and located in the north were favorable for Zygodacus cucurbitae, and that this species was causing the damages in the field. So it confirms that this species is an invasive and aggressive species. Then the second important result is that in the less frequently treated plots, there were more Dacus bivitatus, or these less frequently treated plots allow the outbreak of Dacus bivitatus, who come when the plots are already infested, and also in the fruits that are already infested by other species. So it confirms that this species is an opportunistic species. And finally, we didn't find precise variables that had an effect, a significant effect on Dacus ciliatus as for the other species, so which confirms that Dacus ciliatus is an ubiquitous species in the field, but at the fruit scale, it seems to better thrive in zucchini, young and curved fruit. So that confirms also that it's a species under concern in Africa and in India where it's predominant. And the problem is that to date we don't have a specific or an efficient attractant for this species, whereas we have some insights of attractants for Zygodacus cucurbitae. And finally, the last but not least important result is that we've seen that young fruits are favorable for all the development of all species, which is really here the problem of this pest. And that it highlights that it's crucial to find efficient and a specific control method. I should say multi-specific or pluri-specific control methods. And also it highlights the importance of instructing the farmers about the pest life cycle and the importance of sanitation and prophylaxis measures to reduce the population levels in their fields. So I thank you for your attention, and I thank you also to all the people that helped me during this field work.
Still mine.
Thank you, Louise. Do we have any questions for Louise from the floor? There's one over here.
Okay, thank you. That was a lot of information. So I missed a little bit your argument why you think that some of the species may be competing. So could you please repeat your arguments for that?
We found at the fruit scale that fruits were infested by could be infested by different species, and that we've compared the number of individuals in fruit that were only infested by one species, by Zygodacus cucurbitae only and by Dacus ciliatus only, and we compared with fruits that were infested by the two species, and we found a negative correlation between the number of individuals of Dacus ciliatus. The more Dacus ciliatus there were in the fruit with the Zygodacus cucurbitae, the less Zygodacus cucurbitae there were in the same fruit.
Thank you.
Sorry. Thank you. Do you have any further questions? One on that side there.
Thank you, Louise, for a very interesting talk. You talk about young fruits. How young? Because, you know, Dacus is known to even attack flowers. Now, the female flowers often have this small fruitlet already at the base. So how far or how early did you go in your sampling of young fruits?
Yes, so we did the samplings only at the fruit stage. We did not visit, we did not sample the flower. And I decided that young fruits were fruits under 100 grams and under 10 or 12 maximum centimeter long. Then I said that the next, the bigger fruits were immature, and over 400 grams, I said they were mature.
And so you got a very high spread of the numbers of flies that you get out of it. I mean, was there any correlation there about young fruits having very few pupae? Because, you know, they can't grow anymore, but they probably will not be infested at a later stage because the skin will be too hard.
Yes, but that's what we found. We found the negative correlation between the weight of the fruit and the number of pupae, which is surprising. But it shows that, yes, flies better develop or prefer, either females prefer to lay eggs in small fruits, and larvae, it's also a good substrate for larval development than a mature fruit, because mature fruits, yeah, as you said, the skin is maybe harder to lay eggs.
Okay, thank you. Are there any more questions? No, I don't see any more hands, so I think we can move on to our next speaker. Thank you, Louise.